The overall goal of this procedure is to efficiently grow single-lattice diffraction quality crystals of recombinant proline-proline endopeptidase 1, or RPPEP-1, protein. Without this method, RPPEP-1 instantly produces highly intergrown crystals not suitable for X-ray diffraction analysis.
The main advantage of this technique is that a high number of well-ordered and well-diffracting crystals for structure determination of RPPEP-1 can be produced in a short period of time and with limited material input. This procedure makes use of the microseeding method and can be adapted for every successful initial crystallization condition of RPPEP-1, as well as its substrate peptide complexes.
This method can be adapted to produce well-ordered crystals of virtually every protein that yields intergrown crystals, and can also be used in cross-seeding experiments with protein variants or in co-crystallization experiments with small molecules.
Visual demonstration of the handling of crystallization methods is crucial, since even small variations can have a significant impact on the diffraction quality of the crystals.
Carry out crystallization trials in the sitting drop format using standard commercially available screens and a crystallization robot. First, concentrate the purified protein to 12 mg per milliliter using a centrifugal ultrafiltration device in 5-minute intervals at 4000 times G and 4 degrees Celsius. Mix the protein after each interval to prevent precipitation and aggregation. Determine the protein concentration at 280 nanometers